So today I'm going to talk about our favorite transcription factor, which is estrogen receptor in breast cancer, and talk about a new project that we've just wrapped up. I'm going to start by giving a little bit of an update on a project that's been going on in the lab for a long, long time, something that's really been keeping us busy for a number of years. So we're interested in estrogen receptor, mostly because it is the driving transcription factor in 3 out of 4 breast cancers. And we try and understand this transcription factor and the function of this transcription factor by looking at where in the genome it interacts, because these are the switches, and how does it actually get onto the DNA or the chromatin. Now we know estrogen receptor uses a slew of associated proteins to open the chromatin, to access the chromatin, and to be able to elicit gene regulation, whether that's up or down. And we've been very keen in trying to identify what these associated proteins are. And ultimately, we're really keen in trying to identify the commonalities and the differences in drug-sensitive versus drug-resistant contexts, really with the idea of trying to identify key nodes or molecules that might be of therapeutic relevance. So a number of years ago now, we were using chip-based, so chromatin IP-based methods to try and identify binding sites of estrogen receptor. This was pre-sequencing days. This was microarrays, so chip on chip. Of course, this has been superseded by ChIP-seq now. And what we were doing was mapping where in the genome estrogen receptor was residing. So where in the chromatin was it binding. And these experiments were really quite informative because they taught us a couple of things about estrogen receptor that was new at the time. So one was that estrogen receptor regulates target genes from these distal regulatory elements, which we know is very common for transcription factors now. We didn't at the time. That was new information for us. And by mining these distal regulatory elements, we could look for enriched motifs. And that pointed us towards 2 family of transcription factors, the GATA transcription factors and the FoxA1 transcription factors. And we identified the particular family member within each of those transcription factor families that was relevant. It was GATA3 and FoxA1. And actually, we've done some high resolution transcription factor mapping using a modified ChIP exonuclease approach with Milner sequencing, and allowed us to identify with really quite high resolution how these 3 proteins fit together on the chromatin. And there appears to be predictability in the architecture. So these 3 proteins don't randomly squeeze together on a regulatory element. There is predictability in the spacing. And presumably it's the spacing of these motifs in an open region in the genome that allows these 3 proteins to come together, and that will become an ER regulatory element. And no surprise, these 3 proteins are really at the core of the estrogen receptor complex, the 3 defining transcription factors that make up luminal or ER-positive breast cancer. And presumably, because if you don't have expression of all 3, you don't have a functional estrogen receptor pathway, and you don't have an ER-positive breast cancer. Now, the 3 proteins contribute quite differently to the transcriptional complex. So we know a lot about estrogen receptor. It's the ligand-activated nuclear receptor. So it's a transcription factor that's activated by estrogen, of course. But we can block that ligand binding domain using drugs such as SERMs or SERDs, which are degraders of estrogen receptor. Or we can deprive women of estrogens using aromatase inhibitors. So we can actually block or modulate therapeutically estrogen receptor activity by influencing this pathway. Now, these other 2 transcription factors are a little bit more complicated. So FoxA1 is what's called the pioneer factor. So it has the capacity to bind to compacted chromatin on its own, open that chromatin, and essentially sit there as an adaptive protein to allow other transcription factors to come into that particular region in the genome. Very few proteins have genuine pioneer factor activity, and really the classic pioneer factor is FoxA1. We think it's really more of a tethering mechanism, but it is the foundation protein that actually opens a region in the genome and actually demarcates that as a putative enhancer regulatory element. Now, GATA3 is the 3rd member of this complex, and it's a tricky protein. We've studied it for a number of years. We know it's important. It seems to play quite a complex role, and it doesn't have any really clearly defined function other than maybe to function as a rheostat to increase or decrease the activity of this complex. We've got some unpublished data where it recruits TET2, and it might be involved in DNA methylation there, but it's really has been the odd one out, and we've spent years characterizing it, and we've spent years frustratingly trying to figure out what it's doing. But we know it's important, and yeah, we've just got to get to the bottom of it. Now, as Alison alluded to, we've set up a company called Azaria Therapeutics, and that was really around the finding that this pioneer factor FoxA1 is critical in drug-resistant contexts. So you can get mutations in estrogen receptor, you can get mutations or changes in the stoichiometry of associated proteins, but in all of these resistant contexts, the dependence on FoxA1 is retained. And so this pioneer factor is critical, even in the multitude of different resistant mechanisms that we've looked at, making it an exciting drug target. And this is something that we're going to be actively pursuing in the years to come. Now, one thing we noticed a number of years ago now, when we were applying our ChIP-seq, our transcription factor mapping approaches to primary tumors, and we believe this was the first time that anyone had mapped a transcription factor in solid tumors, we found that estrogen receptor sits on the genome in tumors from women that have different clinical outcome, but it sits in different regulatory elements. And I guess this is no surprise because we know that these tumors express different genes, but what this suggested was that estrogen was involved in both contexts, it just different enhancers. And so the simple way of visualizing it is the estrogen receptor will get recruited to a set of enhancers, we'll switch on a set of genes, and that will confer drug sensitivity to that woman. Now we know, and that will be the platform where cofactors are recruited. Now, importantly, we know this whole complex can move from one set of regulatory elements to a different set of cis regulatory elements. And this is really important. This might not sound really profound, but actually the consequences of this can be really quite dramatic. And we know that if estrogen receptor is regulating this set of genes, this woman is likely to have a better clinical outcome. Whereas if estrogen receptor moves to these different regulatory elements, it's going to regulate different target genes. So we know these genes and their binding profile are linked with decreased response to estrogen receptor targeted agents and decreased overall survival in women. So really trying to understand this mechanism has been a critical question for us and has kept us busy for a number of years now. And what's interesting is the complex doesn't just randomly move around the genome. It appears to go from one set of FoxA1 sites to a different set. And so really this has been a million-dollar question for us. I'm trying to figure out what allows estrogen receptor to go from one set of Fox sites to a different set where it's regulating different genes and the fate of that cell and that woman is distinct. And we rationalize that if it was a movement or a reprogramming of estrogen receptor, so if something coaxes estrogen receptor to move from one set of regulatory elements to the other, then in theory we could push it back, and maybe we can actually push estrogen receptor back to these good outcome binding sites and actually change the outcome of that woman and a treatment response. And actually we found a mechanism, and I've talked about this a number of times over the last couple of years, so I'm not going to show you any of the old data, just give you a little bit of an update. And we found that one mechanism that allows estrogen receptor to move around the genome is another transcription factor called progesterone receptor, which is another nuclear receptor. It's another ligand regulatable nuclear receptor. It's a cousin of estrogen receptor, and there's a long and complex and at times confusing literature between estrogen receptor and progesterone receptor in breast cancer. But what we found was that the progesterone receptor, when it's bound to the DNA, when it's activated by its ligand, it can bind to the DNA and it holds estrogen receptor in these regulatory sites. And so it's really this functional interplay between these two transcription factors that can allow estrogen receptor to sit at these regions. Now, we actually discovered progesterone receptor as a molecule involved in the estrogen receptor complex using a method called RHYME. So RHYME is essentially, it's a fancy IP mass spec, but there are a couple of tricks about RHYME that make it really quite powerful. So the big thing about RHYME is it's endogenous. So it's essentially a modified chromatin IP. So instead of pulling out a tag protein, we're pulling out endogenous complexes. And we've modified it to include tandem mass tag labeling. And this is the new iteration is called QPlex RHYME. So this allows us to pull out protein complexes, endogenous protein complexes, and use mass spec to identify what is actually in that complex, both proteins known and unknown. And this is how we discovered progesterone receptor. Now, this is just one little snapshot of some of the data. This is published a couple of years ago, but this is probably the simplest experiment for us, where we've purified estrogen receptor complex from a cell line model, and our control is IgG. So enriched left of 0 is IgG, enriched right of 0 is ER interacting protein that comes up in multiple replicates. And you can see there's a clear recruitment of proteins to estrogen receptor. And highlighted a few of these, but really these are the cofactors and the chromatin regulatory proteins that us in the field have been piecing together for a number of years now. And we managed to get them in one experiment using this QPlex RHYME approach. Now, the benefit of QPlex RHYME is that because it's endogenous, we don't have to tag our gene. This works from PDX tissue and it works from clinical samples as well. So I'm showing you some data. So this is some data from a number of primary ER-positive breast cancer samples where we split the samples and we purified either estrogen receptor or IgG from the same sample. And we only plotted proteins that came up as a physical ER-associated protein in all of these samples. And it was 5 independent samples from 5 independent women. And again, we capture all of the known associated cofactors and transcription factors, including touch on in a second. Now, what we've done, we've taken the QPlex RHYME and we've asked the question, what changes in the ER complex when we look at ER positive, PR positive cases, and we compare that to ER positive, PR negative cases. Now, we know women that have a tumor that expresses both ER and PR tend to have a better outcome, and they're likely to respond to endocrine agents, so drugs that target estrogen receptor. And conversely, women that have a tumor that by IHC is ER positive and PR negative tend to have a worse outcome and tend to respond less well to these ER targeted agents. So we purified estrogen receptor from a number of ER PR positive tumors or ER PR negative tumors and looked at the changes. And we're only looking at the changes that occur in one context versus the other. And we expected to see a number of differences, but we saw very, very few. And actually, when we looked specifically at transcription factors, because these are the proteins that can change estrogen receptor binding, there was really only one, and that was the progesterone receptor itself. And that was, of course, enriched in the ER positive, PR positive contexts. So the way we rationalize this is between these tumors, you don't see wild changes in the protein complement. So there's not that you're getting major shuffling of the cofactors and associated proteins. It's just the whole complex goes en masse from one set of sites to another. But clearly the one big difference is the progesterone receptor. And so we come back to this schematic or this model where actually one of the main mechanisms that allows estrogen receptor to move from one set of sites to another is the progesterone receptor. And it's the loss of progesterone receptor that we believe allows this complex to move to a different set of regulatory elements and then change the target genes of the ER complex. And actually, this process happens naturally. So this is some data from CRUK looking at breast cancer incidence relative to age. And you can see, of course, as we know, this disease tends to affect older women. Now, if you overlay the average circulating levels of progesterone on this graph, this is what it looks like. Now you can see, this is not definitive evidence, but clearly as native or endogenous progesterone declines, we see an accumulation of breast cancer. And our argument and our hypothesis is that it's this natural decline in progesterone where you lose that molecular repressive mechanism where the progesterone receptor is no longer able to bind DNA and it's no longer able to function in the sequestration mode to change or reprogram estrogen receptor binding. So if that's the case, and if the hypothesis is right, then in theory, we should be able to add back ligands, so either progesterone or a man-made equivalent, a synthetic progestin, and we should be able to reprogram estrogen receptor back to these sites. And in fact, that's what happens. And we've seen that in cell line models, in tumor explants, and we've seen it in PDXs, and I've got some data from some primary samples in a slide or two. Now, based on this data, we teamed up with Richard Baird and Sanjeev Kumar to set up a trial called Pioneer. Now, Pioneer is, I'm sorry, this is quite a complex slide, but Pioneer is essentially aiming to translate the preclinical findings we made around progesterone activity in ER-positive breast cancer. And it's to ask the question, if we combine a standard of care, an ER antagonist, with a progesterone receptor agonist, can we change both estrogen receptor transcriptional activity and where it binds in the genome? So can we actually hit this complex by two different mechanisms? And so the trial has got three arms, an aromatase inhibitor or letrozole on its own, letrozole plus low-dose megestrol acetate or Megase, which is a synthetic progesterone, it's a progestin, and the third arm is letrozole plus high-dose megestrol acetate. And the high dose we know on its own has anti-tumoral activities. So 160 milligrams of Megase is an effective breast cancer treatment in some women. And in fact, in old literature, compared head-to-head, it was as effective as tamoxifen as a breast cancer treatment. And it's really fallen out of favor because we didn't understand the mechanism, and we have drugs that target estrogen receptor directly. But we know this agent can be efficacious. 160 milligrams also has weight gain problems. Now, lower dose of Megase doesn't have weight gain problems, but we don't know whether it has any anti-tumor activity, and that's really the goal of this trial. Now, the trial is a window of opportunity trial, so we'll be collecting samples pre and post treatment, and we'll be measuring endpoints of proliferation, apoptosis, but we're really interested in understanding what happens to estrogen receptor on the genome. And I've got one slide with some preliminary data. Now, we're excited by this because this is an old drug. It's a cheap drug. We know quite a lot about this drug, so really it's about repurposing a safe, cheap drug. And one of the issues in breast cancer treatment is compliance. So a number of women don't complete their recommended treatment course. That are dealing with side effects which compromise their quality of life. Now, side effects from aromatase inhibitors include rheumatoid arthritis problems, but also hot flushes. Now, women have stopped using treatments to minimize hot flushes because they've been told that these agents are quite deleterious. Actually, one of the best things to minimize hot flushes is a progestin, and they're just not being used. And I think that's for another conversation, or maybe for discussion on Thursday. This is in large part because of some data from the hormone replacement therapy trial, which is tainted the view of progestin. So we're hoping this trial will actually clear up some of these misconceptions and will improve the number of compliance and potentially also improve the anti-tumor activity in women with primary ER-positive breast cancer. Now we've just got one slide with a little bit of preliminary data. So these are some patient samples where we've actually got a patient, we've taken a pre-treatment sample and a post-treatment sample. So post-treatment given an aromatase inhibitor plus a progestin, and we've mapped by ChIP-seq estrogen receptor and progesterone receptor, and we can see exactly what we saw in our cell lines, in our xenografts, and our PDXs, where in post-treatment, if these women are given a short dose of progestins, we can recruit PRs to the chromatin and we get estrogen receptors to the same sites. This heat map is essentially showing ER is getting pushed to these sites in the presence of progestin, and it's exactly where progesterone receptor lines up. And we see it in this other patient as well. So even in these patient samples, we can reprogram estrogen receptor binding by adding a progesterone receptor ligand, which validates some of the preclinical data that we showed previously. Now, this is the data I was showing you before. These are our rhyme interactome or rhyme interaction proteins. So this is from the primary tumor data. We get lots and lots of ER-associated primary tumors. Now, we believe a lot of these are just going to be passenger proteins. A lot of them might be functional. So we've coupled this data with some of our CRISPR screening data to identify what proteins do we get as reproducible ER physical associated proteins. Actually, but also hot flushes. Now, women have stopped using treatments to minimize hot flushes because they've been told that estrogen receptor, where the gene is required for viability of breast cancer cells. Now, I'll use this as a segue to talk about just the last part of the talk. And one of the proteins we consistently pull up is a protein called STAT3. Now, STAT3 has a long history in cancer. And we've never really looked at it from our lab's point of view, and mostly because it was a pretty crowded field. But we kept on pulling it up in our rhyme experiments, and had a very talented postdoc who was invested quite heavily in this. I'm going to show you some of his data. Actually, I might get my son to stop that sound the basketball as well. Sorry about that. Now, as I was saying, Rasmus was a postdoc who came to my lab, and so his background was in type biology, and came to cancer epithelial transcription factor, and he was very interested in using his experience and coupling it with ours. And the question for him was really trying to understand the communication between these cytokines that come from adipocytes and the communication with the positive epithelial cells. And he focused on one particular cytokine. Now, we know there are lots of cytokines. Now, this is the data of interactome or rhyme interaction proteins. So these are tumors. Now, we believe a lot of these are just going to be passive CRISPR screening data to identify what proteins often really stood out for the in front of the numbers, about 150. The cytokine comes from adipocytes. I'll use this as a segue to talk about just a lot is a protein called STAT3. Now, STAT3 has a long history in cancer, including breast cancer. We looked at the function of IL-6 that we see in this other patient as well. Now, as I was saying, Rasmus was he came to using his experience. And we know for quite a long time now that women that have high circulating levels, I believe a lot of these are just going to be passenger outcomes. So this is a Kaplan-Meier curve showing women that have the highest. And you focus on IL-6, and there's IL-6, and you can see the outcome is actually across the numbers about a third of the interleukin-6 or IL-6 scans. Now, we use this as a segue to talk about just last about how this pathway works. So IL-6 activates a number of proteins. Cells, they activate an internal cascade of signaling events that include JAK kinases, and ultimately this results in phosphorylation of STAT3 and dimerization of STAT. Just as a reminder, we know in the interleukin-6 or receptor target cells, they activate it, and ultimately this results in phosphorylation of STAT. Quite a long time now, the women that have high STAT3 that has linked functionality, more than half of cases will express phospho or active STAT3. So this is clear positive breast cancer. Now, people have looked for correlations between phospho-STAT3 and outcome in ER-positive breast cancer patients, and not really seen a lot. And I think it's complicated by the fact that STAT5 is also expressed, and that functions to oppose the activity of STAT3. But what Simon Johnson has done is he's gone back to some pretty old samples, and he's looked at the data, and he's stained for IL-6 and phospho-STAT3. And if you look specifically at the women who express both phospho-STAT3, but also IL-6 expression, you can see these women have a worse outcome. So it's really about just stratifying women in the right way. Now, we know that phospho-STAT3 is being linked with metastasis. I'd say The data, probably the most convincing data I've seen is the ERB2 positive tumor model, where we know that ERB positive mammary tumors will occur. If you cross them with STAT3 knockout, you get primary tumor formation like normal, but you just don't get metastasis. So data suggests, this and a number of other data sets suggest that the role of phospho-STAT3 in breast cancer is really linked with cell invasion and metastasis more than primary tumor growth. Now, we wanted to model this in our ER positive luminal cancers, and this is typically quite tricky. Now, a number of labs can implant xenografts, PDXs, and get them to metastasize. We can't. Now, there is a modification on the xenograft approach that improves the metastatic potential of ER-positive cancer models, and this is called the introductal model, or the MIND model. And essentially, the way it works is instead of implanting cells in the usual place, what you do is you actually inject them past the nipple into the duct. And if you do this, the cells will faithfully recapitulate what you see in women. So you get metastases reproducibly to the liver, the lung, the bone, and the brain. Now, we wanted to ask, can we assess metastatic potential in cells that either have IL-6 or are not exposed to IL-6? Now, we looked at this from a number of different angles. It's complicated by the fact that mouse IL-6 does not cross-react with the human receptor, and the half-life of IL-6 is incredibly short. And we made IL-6 pellets, which didn't work. We have no explanation why. So what we ended up doing was making isogenically matched cell lines that express either empty vector or IL-6. And this might seem a little contrived, but actually this is what happens in triple-negative cancers. So we know in triple-negative breast cancers, they have an autocrine production of IL-6, whereas in ER-positive tumors, they tend to get it from paracrine sources. And then we did the experiment, did the introductal tumor experiment. We looked at the primary tumors. We saw activation of phosphostat, and we saw increases in primary tumor formation, and we didn't really expect this. We were looking mostly at metastasis, but we did see increases in primary tumor formation in cells that were stably overexpressing IL-6 versus the control. But when we looked at the metastases, we saw that the cells that were overexpressing IL-6 had much more bone mets and lung mets. So it's really confirmed to us that in this in vivo introductal approach, we can reproducibly map luminal metastatic potential, and we can assess whether IL-6 has an impact, and it does. It promotes metastasis to specific organs. Then what Rasmus did, because we're a functional transcription factor lab, we went back to some of the models in the lab to try and figure out what was going on. And there's lots of literature suggesting that STAT3 and estrogen receptor could functionally interact. We found them as physically associated proteins, so this made sense. But what he did was he went right back to basics, and he took a cell line model and he stimulated with IL-6. We got activation of phosphostat. And then he did an ER Q-plex rhyme experiment. And really the goal of this experiment was to purify estrogen receptor and ask the question, when we activate STAT, and STAT3 presumably comes onto the complex, or goes onto the complex even more than it does in basal conditions, what happens to the ER complex? And again, I assume there was going to be lots of changes, and there weren't. There was really only a handful of changes. And when we activate cells with IL-6, really the main protein that gets recruited to the ER complex is STAT3 itself. And we could validate this in primary samples, because we could show this interaction between STAT3 and ER in both primary tumors and in pleural effusion. And then what he did was what looks like a complex set of ChIP-seq experiments, but I really just want to make a couple of points here. So he mapped phosphostat and STAT3 in this breast cancer cell line. And really there's a couple of take-home messages here. One, if you get STAT3 binding, you always get phosphostat binding. So these two variables correlate almost perfectly. And where you see STAT3 or phosphostat3 binding, you almost always find FoxA1 and in some cases estrogen receptor. So this complex, when it's activated, doesn't randomly go to the genome. It goes to sites where the pioneer factor FoxA1 is. And in a number of cases, you also see co-occupancy of estrogen receptor. And so this made sense because we can capture these in the same complex. So the fact that they got recruited to the same sites in the genome wasn't overly surprising. What Rasmus could do was he could validate this in primary tumor samples. I'm showing you data from 2 primary tumors here, but we see the same thing, where if we take a primary tumor, we map phosphostat or estrogen receptor, and we see really good co-occurrence of binding. And in fact, in this particular tumor, it's about 98% of the phosphostat sites are also estrogen receptor and FoxA1 binding sites. And this is really staggering overlap. This is from a little chunk of primary tumor from a woman who had primary breast cancer. We rarely see this degree of overlap, even in cell line models between replicates of the same thing. So it suggests that this protein, when it goes to the chromatin, tends to go where ER and FoxA1 are already bound. Now, just because it's binding doesn't mean it's active. So Rasmus did the experiment where he stimulated cells with IL-6, and then he mapped H3K27 acetylation. And when we look at the enhancers that become active, and only specifically look at those sites that become active in the presence of IL-6, we can see this is really where all the activity is going on. We get lots of STAT3 at these sites. We get ER is already there, FoxA1 is already there, but we actually get more. So by stimulating STAT3, it goes to the ER FoxA1 sites, and in fact, if anything, we have more of estrogen receptor binding and more FoxA1 binding. And this really suggested there was a clear functional interplay between these two different transcription factor pathways. And then he did this experiment. This kind of threw us. So he generated an IL-6 signature. So he did this in a cell line. In this case, it was T47Ds. Generated that signature, and then he asked the question: if we look at this IL-6 signature, can we see or assess or show that these genes are dependent on ER and or FoxA1? And it turns out they're not. So if we look at these genes, they're completely dependent on STAT3, no surprise. But if we inhibit either estrogen receptor or FoxA1, we see no change in these IL-6 stimulated genes, suggesting that even though these transcription factors go to the same region in the genome, the ER and the FoxA1 doesn't contribute to transcriptional activity. And this was really surprising because we know that most of those regions are only open and accessible to transcription factors because of FoxA1. And we're still working on this, but we've got a working theory at the moment. And he validated this in a second cell line. So he took MCF7, so it's a slightly smaller signature. He has about 70 genes. And in the MCF7 context, he made STAT3 deleted lines using CRISPR deletion, and the IL-6 signature is completely gone if you delete STAT3. But if we inhibit estrogen receptor using, in this case, instead of knocking it down, we've used a SERD, so fulvestrant, we've degraded estrogen receptor, you see no changes at all in the IL-6 signature, suggesting in the second model that the genes that are switched on by IL-6 are completely independent of estrogen receptor. And in fact, he looked at the binding in the presence or absence of FoxA1, and phosphostat binds perfectly well, even when FoxA1 is depleted. Binds exactly the same with the same affinity to the same sites. So this was really puzzling for us. These transcription factors, STAT3 converges on ER FoxA1 sites, and yet you can take ER and FoxA1 out of the equation and STAT3 still functions. And you know, we were a little perplexed by this, but we saw the same thing in patient samples. We've got two data sets. I'm only showing you data from one, but the other data set looks exactly the same. So these are biopsy samples from women pre and post an ER targeted agent treatment. So samples were collected pre-treatment, and then women were given an aromatase inhibitor, so letrozole, for at least 4 months, and then biopsies were collected at the end, and gene expression was conducted. Now, if we look at our IL-6 signature from either of these cell lines, they are not affected in these patient samples at all. It's basically background. Whereas the ER target genes that we know and love, that are consistently regulated by ER, and FoxA1, we can see that these decrease in these women. So this really supports the notion that these target genes are completely independent of ER. So if they're resistant to estrogen receptor targeted agents, we were looking for something that would block activity of the STAT3 pathway. The obvious one was JAK inhibitors, because this has been shown in the literature in other contexts. Now, so to assess this, Rasmus set up an explant or a tumor ex vivo experiment where we took a PDX tumor from Carlos and Alex Brunner, and we could cultivate it on these dental sponges, and they're viable for about a week. So we can do treatments of the same bits of tumor, and we can look for proliferative responses. And if you treat with this compound called ruxolitinib, which is a JAK1, JAK2 inhibitor, so that's really the protein that activates STAT3, we see complete loss of phospho-STAT3 and activation and decreased proliferation. And if we quantify that, the numbers are shown down here. So we can block phospho-STAT3 activity, just not using an ER-targeted agent. We need to block this particular pathway. And if we do that using this ruxolitinib, we can block phosphostat activation, and we can block the proliferative potential of the tumor. And we've looked at a number of target genes of IL-6 signature genes, and we see these down as well. And then finally, we wanted to look in resistant context. So we've taken a PDX, and again, this is from Carlos. So this is a PDX for a woman that was resistant to SERM, so to tamoxifen, resistant to CDK4/6 inhibitors, resistant to all of the ER-targeted agents. And we did the experiment where we treated with either fulvestrant to block estrogen receptor, to block this phosphostat3, and then we looked at the tumors. And we didn't see much of a difference on primary tumor growth, but when we looked at the invasion of these tumors, and we measured this by actually looking at the tumor cells leaving or breaking through the myoepithelium, breaking through the basement membrane, we could quantify that. We say, actually, if we look at the invasive potential of tumors in either control or our ER-targeted agent treatment, we see that actually these are still invading. They're busting out of the tumor compartment, and you can see they're really raggedy. You can see them here and there. They're obviously on the move. Whereas if we block the phosphostat using this ruxolitinib, these tumors stay self-contained, and we can quantify that, and the invasive area goes down dramatically. So, preliminary data, but it suggests that if we block the JAK pathway, we can stop the invasive and metastatic potential of tumors, even if they're resistant to ER-targeted agents. And now, interestingly, there is a clinical trial that's running from UPenn, which is assessing the combination of ruxolitinib with an ER-targeted agent in a metastatic context, and that's, as far as I'm aware, has finished recruiting, and so we were obviously keeping an eye out for that data. So just to sum it up, so there's a couple of transcription factors that converge on the same regulatory elements. The upstream stimulus for estrogen receptor is estrogen. Stimulus for STAT3 is IL-6. We know other ligands can, of course, activate STAT3. What really threw us for a long time was these transcription factors converge on the same regulatory element. So STAT3 goes the way ER and FoxA1 is, but it essentially hijacks these regions, and it uses these regulatory elements, but in a manner that's completely independent of ER and FoxA1. And what this suggests is that the drugs that we use to block this pathway will not cross-inhibit the other transcription factor. So drugs that block estrogen receptor will block estrogen receptor and only estrogen receptor, and this contributes to primary tumor growth. And actually, drugs that block STAT3 will block specifically this transcription factor arm of the axis, and this will block metastatic potential of that primary tumor. So finally, I think I mentioned people along the way, but really all the last work I spoke about, the IL-6 work, was really all the work of one fantastic postdoc called Rasmus Siersbeck. We have really fantastic computational support. Eva was really driving the Qplex rhyme and has been involved in every experiment using Qplex rhyme. Really fabulous collaborators. And thank you very much. And I look forward to having some questions on Thursday.